All right, is everybody ready to get started for this next session? So now we're going to move on. I think it was a great, introduction earlier with a lot of the data blitzes, and now this will be a really exciting session, really talking through, we've heard a lot about some of the basic mechanisms and linked to disease, and this is going to focus on what's going on in terms of clinical trials targeting the GBA pathway. So we'll get a nice overview from Simon in terms of the current status of things, and then throughout the rest of the session hear updates from venglustat, from Embroxol, and gene therapy-based approaches. So we'll start with Simon Stott's presentation, entitled The Landscape of Current Preclinical and Clinical Programs.
Very good. So firstly, I'd like to thank Ziv and particularly Lorna, wherever she is, for organising such a fantastic event. Cure Parkinson's is very proud to be one of the sponsors, and thus far, I think it's really been a spectacular meeting. So my only disclosures is that I'm an employee of Cure Parkinson's, a small research charity focused on disease modification for Parkinson's. And each year at Cure Parkinson's, the research team puts out an annual report in which we look at the landscape of the drug therapies being developed for the condition. And we do this with fantastic support from the Journal of Parkinson's Disease, firstly, but also from a group of Parkinson's research advocates, Kevin McFarthing, Sue Buff, Gary Ravaloff, and Ken Bitzer. And we couldn't do it without, I couldn't find a picture of Ken. And we couldn't do it without support, the expertise of Brian Fisk from the Michael J. Fox Foundation, another valuable member of the team. But when I was asked by the organising committee, which included myself, strangely, to give this presentation, I thought, how do you cover, in the space of 15 to 20 minutes, the entire landscape, preclinical and clinical research? But I thought that this report could offer sort of a structure. And so now you've got to give me a lot of rope here, because this was a 5-minute analysis, real back-of-the-envelope stuff. But I went into the clinicaltrials.gov website. This is what we do with our Parkinson's report. And I downloaded all the data for active clinical trials, looking at an intervention with keyword Gaucher and GBA. And it came back with, when I cleaned it up a bit, it came back with 29 studies. And there were a number that had no phase indication, but what was quite pleasing to see was the general spread of clinical trials across the 3 phases of clinical testing. Here in phase 3, you've got 2 venglustat studies, and you've got the Embroxol study, and you've got several ERTs. And also a good spread across the 4 main fields, or the 4, classes of therapeutics for GBA approaches. The chaperones are dominated here by Embroxol. There's 8 of the 9 studies involving Embroxol. And gene therapies are really coming through. Now, again, just be careful with what you take from this, because there are a lot of studies that aren't included. For example, there are a number of studies registered on other clinical trial registries, such as in China, for example. There's quite a few studies coming through. And if the keyword Gaucher and GBA were not mentioned in a phase 1 study looking at healthy volunteers, then those dropped out as well. But when I started out with this effort, with this project, this gave me some structure to apply to the talk. And so what I'm going to do is just give you a very brief view from 30,000 feet of some of the activities looking at these 4 areas. We'll start off with enzyme replacement therapy. So in the early 1990s, intravenous ERTs were introduced with Algluceras, and one of the challenges with this approach was the starting material, human placental. It was to supply a worldwide market, you needed basically 10 metric tons. And so in 1994, recombinant versions was introduced. And with great success, within 4 years of initiating therapy, more than 90% of patients were showing significant improvements. And at the moment, there's basically 3 main ERTs that are applied for Gaucher's type 1. The treatment regime is typically every 2 weeks infusions. Immunogenicity is an issue, with about 15% of patients developing antibodies. There's also cost. And the pharmacokinetics is not perfect either. The half-life is very short, only about 30 minutes, and there's no CNS penetrance. And the CNS penetrance detail is quite apparent in the current preclinical landscape. A lot of the ERTs that are being developed preclinically are focusing on CNS access, and leading the way here is Roche and Denali, who have both developed blood-brain barrier transport vehicle technologies. And then we've got the clinical landscape. A couple of interesting studies. One is the Pearl study out of UCSF, which is looking at in utero delivery of ERTs. It's a very small pilot study, just at the phase 1 level. And then we've got the small study in Toronto looking at the use of high-frequency ultrasound to open up the blood-brain barrier in a targeted fashion. Professor Callier was talking about this yesterday. Moving on to pharmacological chaperones. These are agents that can cross the blood-brain barrier and modulate GCase activity or protein levels, as well as translocating it to the lysosome. There are basically 2 categories here. There's inhibitory and non-inhibitory. Inhibitory chaperones have been around for quite a while. They bind to the active site of the enzyme and release it only in the acidic environment of the lysosome. And the early screenings studies that were conducted, the first screening studies basically isolated immunosugars, but these were difficult to translate into clinic because they had a lot of off-target effects, even though they had fantastic results in cell lines carrying GBA variants. And then there was a drug screening study in 2009 which highlighted ambroxol, and an early clinical trial suggested some interesting or encouraging results in Gaucher's type 1 cohort. We've got Stephen Mullen, who's going to be talking about ambroxol, so I won't steal any of his thunder. But just to give you full disclosure, Cure Parkinson's is supporting the current efforts with the phase 3 study. This is a 2-year trial which will involve 330 participants, half with the GBA variant and half without. And we're identifying the GBA cohort through the PD frontline sequencing study. And then we'll move on to non-inhibitory chaperones. These are also known as allosteric enhancers, and they assist the folding of the GCase protein and also help with the translocation to the lysosome. Early molecules that were identified in screening studies include NCGC 758 and 607, which reversed not only inflammation in Gaucher's cell lines, but also reduced alpha-synuclein buildup in Parkinson's dopaminergic neurons. And more recently, Dimitri Krank and colleagues have identified S181 also as another non-inhibitory chaperone. And here the preclinical landscape is quite a crowded situation, with a lot of parties developing new chaperones. Just to highlight here 2 examples, there's Pneumora, which has a collaboration with Amgen. And given that there are people from Congruence in the room, I thought I'd mention Congruence as well. They had a poster here at the meeting. And there's also been some recent drug screening studies which have highlighted potentially repurposable molecules, such as quetiapine. And the clinical landscape for the GK chaperones is really quite encouraging. We now have 3 active programs in addition to the Ambroxol work. We had BL Biotech initiated their phase 2 study in May of last year. Gain Therapeutics is currently in phase 1 testing, as we've just heard, and Vanqua Bio has just initiated this year in April their phase 1 program. I think with all this activity, it sort of reinforces the need for identifying patient-ready cohorts of GBA carriers, and also the need for more standardization of assays and better endpoints to support this clinical trial effort. Moving on now to substrate reduction therapies. These are small molecules that inhibit the biosynthesis of the enzyme glucosyl ceramide synthase, and there's 2 approved. Well, most of the market is down to 2 approved SRTs, which is imglistat and iliglistat, which were introduced in 2013 and 2014 respectively. The clinical landscape here is also focused on, it was initially focused around brain penetrant allosteric inhibitors, but as Pablo is going to talk about shortly, this effort in GBA-associated Parkinson's did not meet its primary endpoint, but is still being tested in Gaucher's type 1 and 2, and as well as Fabry disease, so it's still an active program. And it hasn't stopped others from continuing to develop new SRTs as well. ACE-Link Therapeutics is an example here, which is actively in phase 2 at the moment, I believe. And then there's also an interesting situation where we're seeing the combination therapies being tested, both SRTs and ERTs together in very specific cases. Not all people with Gaucher's disease will require this, but in certain situations this could be potentially very beneficial. And finally, moving on to gene transfer, where we aim to correct a faulty gene or replace it with a healthy gene in order to facilitate disease correction. I'm not sure if you realize this, but there's been this real tsunami for the regulators recently in terms of gene therapeutics. It's really a golden age for this field, with a number of really spectacular results coming through, such as spinal muscular atrophy, and a large number of applications going to the FDA and EMA at the moment for approval. And what's really encouraging is the amount of activity we're seeing in the GBA field around gene transfer. There's a great deal of research looking at, as we've heard this morning from Capsida, around carefully designing the capsids of adeno-associated viral vectors. And we've got a number of companies just highlighted across the bottom here, all actively looking at GBA as a potential indication. And the ones highlighted in red, Lingy Mid and Arvo Bio, are actively in clinical trials. Although Arvo Bio at the moment, just in the last month, they've merged with Tectonic Therapeutics, and Tectonic Therapeutics hasn't been very clear as to what they're going to do going forward. But one good example that we're about to hear about from Sebastian is the clinical trial work that is being conducted by Prevail Therapeutics in collaboration with Lilly. This is where they're using an AAV9 viral vector to overexpress GBA1. They're using both IV as well as delivery to the back of the brain with their Propel and Proceed studies. And one interesting, I was quite intrigued as I wasn't aware of them until I started preparing for this presentation, was Freeline, coming out of Stevenage of all places. They have developed a variant of GCase which is more stable. It's got a much longer half-life, 21 times in the lysosome, and in human serum 6 times more stable. And that's currently in phase 1 clinical trial testing as well. So a lot of really encouraging work being conducted. So just to summarize the main points, my first main point there, a low number of clinical trials was just kind of a controversial statement to encourage more translation through to the clinic. The work is still primarily focused around the four main areas. There's a lot of work being done in terms of CNS penetrant ERTs and new allosteric chaperones coming through, advancing through clinical testing. The ambroxol study is going to get started in Parkinson's for phase 3. There's an opportunity for combination therapies, not just in terms of SRTs and ERTs together, but maybe looking at other opportunities with alternative modalities, maybe anti-inflammatories or a mitochondrial targeting therapeutic potentially. And finally, we've got the gene therapy work, which is quite exciting as well. And I'm not sure how I'm doing for time there, but I think that's my last slide, so I'll say thank you very much.
Time for about one question. All right, I'll ask a question. So Corey, you noted like there's some great opportunities potentially for pre-competitive collaboration with all of the excitement in terms of companies moving forward. What do you think are some of the major areas where there could be a lot of value for companies working together to try and answer some key questions that'll help the whole field?
In the pre-competitive space, we really need to focus on the assays and the endpoints, the clinical endpoints in the studies, just because the patient community is going to look to us at the end of the day and say, what did we learn from all of these clinical trials? And if all we have to say is that this study found this and this study found that, but there's no overlaps between the results, it gives them a very confused message. So particularly around assays and endpoints, we need, because to be honest, another controversial statement, some of this stuff is really all over the field. And we want to be able to do comparative analyses across studies. So yeah, those would be my two sort of
Fantastic.
points to highlight.
That's a very great transition to the next talk, where Pablo will